Okay, so we keep this whole series as anatomy of questions, and here we want to discuss about the detailed structure of a question, because if you understand the structure of a question very clearly, you can answer them fast. Okay, and sometimes understanding the structure is more important, even important than knowledge of the subject itself. So today we will talk about unit 1 and unit 2 questions. Okay, so 6 questions from each unit. Approximate number of questions you can expect from these units in part C in CSIR NET exam. So hello, this is another lecture about anatomy of the question, and here we are going to talk about unit 3 questions. Okay, so let's begin. Let me take a color first, and then we'll begin with discussing different unit 3 questions. 6 questions I have selected, so let's begin with it. Okay, the first one, chromosome organization becomes clearer from a series of biochemical, electron microscopic and X-ray crystallographic studies. When interface chromatin is isolated in low salt buffer and observed under electron microscope, 11 nanometer bead on string organization is seen. Now, can you see where it's growing? Interface chromatin directly observed under EM shows 30 nanometer fiber. When histones are depleted from the metaphase chromosome and visualized under electron microscope, it shows a huge number of very large loops associated with scaffold. Following interpretation can be made from this. Now, so while I am reading this question, right after reading this 11 nanometer beads on string model, I thought that this question is going to be the structures of nucleosome, the structures of chromosome and chromosomal organization. And I am right. The second line states 30 nanometer fiber, then a scaffold protein structure. So this is nothing but the question from the chromosomal structural arrangement. So if you know that, you can attain the question. It's easy. Because every single details are given, 4 statements are also given. You just need to find out the correct combination of statements. Okay, so let's read the statements. 11 nanometer fiber is formed when nucleosome are brought closer by scaffold. No, if you know the structure, you will positively know that this is wrong. You know, whenever you read this question, the first thing that you draw and think of is this. So this is the structure of the chromosome. You know, first the DNA, double helix, then the DNA is wrapped around histone. So when the DNA is wrapped around histone, we call it 11 nanometer beads on a string model. So in this beads on a string model, there is only DNA and only histone proteins. H3, H4, H2, H2B and H1, nothing else. So this statement of A is wrong. So if A statement is wrong, then option 1 is wrong, option 2 is wrong, option 3 is also wrong, because all the options carry option A. So that means the statement B and D in the option 4 is correct. The rest of the statements are not correct. So we can easily find it out within a minute. But again, for your understanding, let me explain all of that. 30 nanometer interface chromatin is formed a zigzag organization of nucleosome of 11 nanometer fiber. So if you look at the 30 nanometer fiber, this is 30 nanometer fiber, and this is the zigzag appearance of the beads on a string model that forms 30 nanometer fiber. So B is a correct statement. C, 30 nanometer fiber makes a solenoid packing to form the metaphase chromosome. No, because the solenoid packing is while at the 250 nanometer wide fiber stage. So this is not the solenoid packing. Okay, so 700 nanometer, 12 like 300 to 700 nanometer, that's the time for solenoid packing. So obviously C is not correct. 30 nanometer fiber gets organized into loops due to SARS getting associated with the scaffold proteins coming closer. That's true, because you know this 30 nanometer fibers, they are getting closer and closer, forming this loop. So the loop is formed with 30 nanometer fiber, and once multiple loops are formed, that forms a 300 nanometer fiber, and that 300 nanometer fiber will start arranging in the scaffold proteins to form the particular final model of chromosome structures at the end. So obviously D is also correct. B and D both are correct. So option 4 is correct. Okay, so pretty easy, right? You need to know one concept and you can answer that. Now let's talk about this one. Amino acyl tRNA synthetase are very specific for amino acylation of tRNAs with the correct cognate amino acids However, there is a possibility of mismatch between the tRNA and its cognate amino acids. So obviously, we know there are this 20 different types of amino acid tRNA synthetase. They are using 20 different types of tRNA for charging. This error is corrected by the inherent proofreading activity of the amino acid tRNA synthetase enzyme itself. In case of two very similar amino acids, namely valine and isoleucine, isoleucyl tRNA synthetase employs the following possible approach for an error-free and amino acylation process. So this is quite interesting problem. CSIR asked this question from this amino acid tRNA synthetase part almost every single season of the exam. So this is a very interesting question, you know. If there are errors in the amino acid, how exactly this amino acid tRNA synthetase is going to find that and remove that? What will be its proofreading activity? This is the question from there. If you don't know about it, you may leave. But if you have the basic idea that the proofreading can be done, first of all, because you need to understand the steps of the amino acid tRNA synthetase. Okay, the steps, there are two-step process. The first step process is association of amino acid with ATP at transfer of the AMP to the amino acid. Then the second step is transfer of the amino acid from there into the tRNA. So if you look at the statements, it removes an incorrect amino acid by hydrolyzing the amino acyl AMP linkage followed by the first reaction step. This could be a possibility. It is activated for proofreading activity leading to the breakage of the bond between the wrong amino acid and the tRNA. This is also a thing. It has an intrinsic ability to recognize the structural difference between amino acids leading to the abortive elimination of the non-cognate amino acid. No, this is not a possibility. There is no intrinsic ability of the amino acid tRNA synthetase to find out the proper amino acid as well as to find out the proper tRNA for the amino acid. It gets sequestered in the second step with the wrong amino acid, and that freezes the amino acylation process. No, this is not even truth, because in this case we are not talking about the addition of multiple amino acids by a same enzyme. The enzyme is specific, okay? But sometimes what happens, you know, the very first step, amino acid and ATP, two phosphate groups are released. ADP is associated, like AMP is associated with the amino acid. That's what we are saying, that it removes an incorrect amino acid by hydrolyzing the amino acyl AMP linkage. This amino acyl AMP linkage is hydrolyzed followed by the first reaction step again from the beginning to conclude the process. And obviously it activates the proofreading activity that breaks the bond between the wrong amino acid and the tRNA in the very second step. So the very first step, the amino acyl AMP linkage is broken, and the very second step, the bond between the amino acid and the tRNA is also broken. So A and B both are true in this case. So option 1 should be the correct one. Okay, so again you see this question is little complex from a mechanism, and that mechanism is the transpeptidation, means charging of tRNA with the respective amino acid with the help of the enzyme amino acyl tRNA synthetase. Let's move to the third type of question. This one is microRNA. miRNA have recently been shown to play a significant role in the fine tuning of the gene expression. Some miRNA induce gene silencing by binding to the mRNAs and inducing inhibition of the translation. On the other hand, there are miRNAs that bind to mRNA and activate their degradation. The following characteristics can be applicable to miRNAs that inhibit mRNA translation. Okay, so some important properties about mRNAs are listed here, and you need to find out the correct combination of properties from there. So when you read this first part of the question, you know, sometimes you don't need to read all the statements. By reading the first part of the question, you understand the topic of the question. You know, this question is from the properties of miRNA. So if you have prepared that, then you attain it. Otherwise, leave these questions. For every single question, read the first part of the question. Do not read the rest of the sentence, because it will be a long time, okay? And you need to think about the statements. So statements are the thing that you need to think about. So if you read the question, if you think like this is beyond your knowledge, you don't know that, you haven't read it, just leave it. But if you read it, then definitely attain it, and then read all the statements. So regarding miRNA, let's say we prepared miRNA, so we know we can answer that. Let's begin. And this question is also from the topic that we already discussed in the class. miRNA is partially complementary to region of target mRNA in the 3' UTR. miRNA always base pair. So think about this first one. miRNA is partially complementary to the region of target mRNA in the 3' UTR. And that is kind of a truth. Okay, if you don't know that, read the next. miRNA always base pairs with the mRNA around the AU rich sequence. We know that this is definitely not true. So B is false. If B is false, then option 1 is not true. miRNA base pairs with mRNA through 6 to 7 nucleotides at a 5' end referred as a seed sequence, as well as a few additional bases elsewhere. Yes, that's true. For miRNA's interaction with the mRNA, so let's say this is the mRNA, this is 5', this is 3' of the mRNA. So in the 5', the miRNA will bind. So the thing is, the miRNA, this is the miRNA, okay? The miRNA binds with this 3', 5' interaction. So the miRNA binds with the 5' of mRNA. This is mRNA, this is miRNA. Now the thing is, this is the interaction in the 5' region known as seed or seed sequence that is present there, okay? And there is no further interaction, no further binding. So those regions are free without any interaction, because the miRNA is not 100% complementary to the mRNA, but it is more complementary in the 5' region. So the first statement, miRNA is partially complementary to region target mRNA in the 3' UTR, is false. They are more compatible, they are more complementary to the mRNA in the 5' UTR rather than the 3' UTR. So A is also false. If A is false, then option 2 is false, option 4 is false. So that only leave us with option C, that is C and D are correct. We know C is correct. Now for D, miRNA is always partially complementary to the 5' UTR. Yes, that is the truth. So C and D both are correct. So you know, if you know 2 of the rest 4 statements that are given in a combination question, you will always end up in choosing the right combination. That's why I always encourage you to pick these combination questions, because these combination questions can help you to look at this. Like these options can guide you to go very close to answer a question, even knowing 2 out of 4 statements. If you know 3 out of 4 statements, you will definitely can answer it. If you know 2 out of 4, you can still answer it. But if you know 1 out of 4, that will be a tough situation for you. Okay. So let's move on to the next question. Attenuation is a mechanism involved in the regulation of tryptophan operon in E. coli. When tryptophan levels are high in the cell, region 2 of the tryptophan L, TRPL, is blocked from pairing with the region 3. This allows the pairing of the region 3 and 4, leading to the formation of row independent termination complex. What would be the structure of the TRPL region in the E. coli cell where the protein synthesis has been initiated already? Now one thing I must tell you, this question when you start reading, you know this is from tryptophan operon and this is from the attenuation of tryptophan operon. We all know about attenuation, we all study attenuation very well, because you know question can come. So when you see this kind of questions, just dig into it and score 4 out of 4. There is no way you can go wrong in it. Now this is only about, like, the attenuation is only about the formation of the stem loop structure, either between 2 and 3 or between 3 and 4. Okay, depending upon the availability of tryptophan. And what we know, if you think about this picture, it will be clear in your mind. So when you are answering this question, flash this picture in your brain, that this is the mRNA. This is the question said that what will happen where the protein synthesis has initiated, and they're supposed to initiate the protein synthesis. The only way attenuation works is when the DNA, when the protein synthesis or translation of the mRNA of the tryptophan operon is turned on along with the transcription. Because of this transcriptional coupling with translation, we have this process of attenuation. And what we can see is that this is the mRNA, ribosome is moving through it, and the ribosome can stall between the sequence 1 and 2 in the tryptophan codons. That leaves sequence number 3 and 4 as separated, 3 and 4 out. This is a situation where the stem loop formed between 3 and 4, which is told in the question itself. 3 and 4 form this stem loop, and this is a inhibitory loop. That means, you know, transcriptional So this loop will not allow the ribosome to move. This is the inhibitor loop. Now normally in a situation where ribosome stalls in 1, where the ribosome, I mean there is very tryptophan is absent, so in that case the ribosome remains in the 1 for a long time because there is no tryptophan. So in the low level of tryptophan, ribosome stalls in the sequence number 1, leader peptide sequence number 1. So as a result of the stalling, 2 and 3 become free. 2 and 3 also have the complementary pairing, so they pair with itself. So 2 and 3 is not a terminator, it's an anti-terminator loop. So this synthesis of protein can continue over the loop as well. It can dissociate the loop and can continue with the translation. So as we know, in this case region 3 and 4 will form this loop. 1 and 2 will be occupied by ribosome. That should be our idea. So region 2 pairs with region 3, allowing transcription. No, it's not truth. So literally if you read this question, they only want to pick up the right combinations from the question's first part itself. So if you read this question for first part itself, you are going to get the answer very clearly, right? Region 1 and 2 will pair, allowing 3 and 4 to pair, leading to the attenuation. Now there is no pairing in the TLPR region. Transcription of structural gene occurs. Region 2 and 3 will pair, leading. So 2 and 3 pairing is never possible. Pairing of 3 and 4 is possible. In this case option 2 states region 1 and 2 will pair. No, they will not pair. They generally are occupied by ribosome. So it's not entirely correct statement. But the second part of the statement, allowing 3 and 4 to pair, leading to the attenuation, that is a truth, and that is already told. Okay, so option 2 will be the correct answer. Okay, so literally if you read the first part of the question, you already know the answer, and that's what you need to pick from the options. So literally these are like melt in a mouth question. Okay, so now let's move to another interpretation question. This is also a very easy one, although people when see this question get very people think it's complicated, but when you read the question it will become really easy. Sometimes we just leave question by looking at the appearance, but actually if you read it, you need only one concept, the concept of transcription, the concept of the presence of promoter during the transcription, to answer this. So in order to identify the regulatory region of a novel promoter sequence shown above, 4 150 base pair deletion constructs were made in a luciferase reporter system as indicated above in the boxes A to D. After transfixion, the observed level of promoter activity in percentage as analyzed by the luciferase assay of all constructs is indicated in the right of the figure. Identify the best correct combination of regions in the options given below that indicates the presence of positive and a negative regulatory elements respectively. That means among the options, the first one will be positively influencing and the second one will be negatively influencing element in the DNA for transcription. Okay, so this is the promoter activity measured as a luciferase activity. So the promoter activity 100% is for control, where there is no mutation done, no sort of mutation. But apart from that, there are 4 situations where deletion is performed, A, B, C, D. So among these 4, deletion of 150 base pair deletion, in all the cases the position of these deletions are different. So in some case we delete this portion, some other case delete this portion, some other this portion, some other this portion. Now depending upon this deletion, the outcome of promoter activity varies. Now we need to figure out which among this DNA element is needed for a positive influence for transcription and which is involved as a negative influencer for transcription. Now think about it. In the DNA, if there is a positive influencer, it will allow the transcription to go fast and more. Okay, so more transcript will be produced. So the promoter activity must be high in presence of the positive influencer. So now if we delete the positive influencer, then the promoter activity should decrease from 100% below 100%. Okay, and if a region of the DNA negatively influence or inhibit a transcription, so normally if we delete that part, then the promoter activity will increase than 100%. So now what we try to figure out is we are trying to see the results where the promoter activity is decreased, the than 100%, where the promoter activity is increased by the 100%. Okay, so the promoter activity decreased to 20% if we delete the gene sequence A. That means this is the portion where positive influencer is in place. See, that's what we saw here. So positive influencer was there. So if we delete that, then the promoter activity decreases. Okay, so A is definitely positive influencer, and among all, 140% means we can see increment of greater than 100% if we delete C. That means, remember, C is a negative influencer. So as C is a negative influencer, if we delete C, we see a huge increment in the promoter activity, because in presence of C, the promoter activity is not that much increased because they inhibit the function. Okay, so that's how you need to figure out. So instead of telling you the function of a positive element and a negative element in the DNA for transcription, they formed a perfect question to trick you so that you leave the question like this. So this is the beauty of the CSIR NET questions in part C. I told you always, they always ask questions from very basic parts, just promoter's role and the presence of positive and negative influencers. But you see the question, the angle of the question is asked in a way involving, cells, luciferase and chemical environment and experimental environment, so that to make you confused. So don't be confused. If you know the concept, look at the question, read the question couple of times, you'll get it clearly in your mind. Okay, so let's move on to the next one. The following statements are made on DNA replication. A replication fork is a branch point in a replication eye or a bubble. A replication bubble contains two replication forks. Now think about it. So when you start reading the statements, you know that this question is not that big. It's small. So less time consuming. And this question is asked from DNA replication, general idea regarding the replication fork. So if you know about replication fork, which you should, you can answer this question quite easily. So replication fork is a branch point in a replication eye or bubble. The statement is true because, if I show you a replication fork, so this is a replication bubble, okay, and in the replication bubble there are two replication forks present in the replication bubble. Okay, so it's replication eye or replication bubble, two forks are present. Replication bubble contains two forks, that's also true. A, B both are true. DNA replication is continuous according to the interpretation made by Okazaki. No, not at all. DNA replication is not continuous in the lagging strand. That is known as Okazaki fragment. So this statement is false. Multiple priming events are required for both leading and lagging strands to initiate the DNA synthesis. Again false. Multiple priming is needed for the lagging strand. Less priming are needed for the leading strand. For one or two, less priming are needed. So this is also false. So A and B are the correct statement. So option 1 is a correct option. This is probably the easiest type of problems you can get. You may not get this kind of easy questions every year. If you get this kind of easy questions, then the cutoff of that year will be high. In order to study the transcription factor TF2H, it was cloned from a large number of human subjects. Surprisingly, the subjects having mutations in TF2H also showed defects in their DNA repair system. So that means TF2H has some role in the DNA repair mechanisms in human. Given below are explanations. Now from the explanation you need to find out the correct answer or correct combination. Let's read it. So this question is from the transcription factor 2H, means eukaryotic transcription. So you may know, you may not know about eukaryotic transcription, about TF2H. If you know, because I showed you already the table regarding all the transcription factors in eukaryotes, and I told you to remember their function. It's very important to know. If you know the function, you can answer this question. And from that function, directly you can choose the correct option without even reading the statements. I can explain that, I can tell that the TF2H is actually linked with a very specific type of DNA repair mechanism. Okay, so let's read the statement. DNA damage is always associated with transcriptional inhibition. Not at all. DNA damage can be done by many other factors. So it's not true. TF2H has no role in DNA repair. False. Because in the question it's already showed, in the experiment, the TF2H also showed defects. I mean, the mutation in TF2H also showed defects in the DNA repair. That means obviously the TF2H has some role in DNA repair mechanism. So B is also false. A is false, B is false. So obviously statement option 1, option 3 both are Wrong. So now the right option is between option 2 and 4. So either C is correct or D is correct. In mammalian system, TF2H plays an active role in transcription coupled DNA repair process. That is what I said. That is the true finding. That is the thing. If you know that, you can select it. Other than that, read the fourth statement. Because of mutation in TF2H, transcription initiation is inhibited and incompletely synthesized mRNA. Mutation in TF2H, transcription initiation is inhibited and incompletely synthesized mRNA remain attached to the template DNA. No, this is not a truth. So there is nothing to deal with, the link of TF2H with the DNA repair. The only statement that explains the relation between TF2H with DNA repair is this statement C, because we know that has some role. So yes, TF2H is actually involved with transcription coupled DNA repair mechanism in mammals, in case of humans as well. So C is correct. So option 2 is a correct option. Okay, so not nucleotide excision repair, but associated with this transcription process, we can see that involvement of TF2H involved in the process of the repair mechanism. Okay, now this one. With an intention to identify the genes expressed in an organism at specific stages of development, mRNAs were isolated from the given organism. cDNAs were synthesized, cloned into suitable vector and sequenced. A few of the cDNA sequences showed to no match with the genomic DNA sequence. Further, it was observed that the sequence were EU-rich and found to be in stretches dispersed along the sequence. The following may be possible reason for appearance of such RNA. Now, what we are talking here, we are talking about cDNA, we are talking about the mRNA. We know if we prepare cDNA, it should have a perfect complementary sequence with the mRNA. And that's supposed to happen, like always 100% complementary feature, but we found out that it's not 100% complementary, it's partially complementary because of some extra added uracil residue, U residue present in those DNA, which is not allowing them to have a 100% complementary nature. Why so? Now, if you know the answer, by now you already know it. We are talking about RNA editing. Remember, in RNA editing process, we have talked about the addition of U with the help of uridylyl transferase 2-tase enzymes to insert U and then sealing the nick with ligase. The 3-step process using endonuclease to cut, right? Then TUTS 2-tase to add U and then to seal the nick with ligase. So this step process can complete the process of guide RNA mediated RNA editing. So this is the only explanation for this process. Now read the rest of the statement and can find the answer. Okay, the statement says that due to splicing, the answer is no, definitely not splicing. Alternative splicing, trans-splicing, and guide RNA mediated introduction of U involving endonuclease terminal uridylyl transferase RNA ligase E. Obviously, D should be the correct option. Only D is the correct option. No A, no B, no C. So they have no role in this process. When we start reading that U-rich sequence are incorporated, the first thing that always should come in our mind is this is all about RNA editing. Okay, so this is how you can solve it. Now, if you don't know about it, so this is the RNA editing process. For your memory freshening, I just put the same picture that we used to describe the process in the lecture. Okay, so this is the steps. Deletion of the breaking of the phosphodiester backbone, then association of U, and then sealing the nick with ligase E, and that's how the process gets ended at the end. So this is how the process works. So again, this is a mechanism based question. If you know, you can answer. If you don't know, then it will take some time, but you cannot answer if you don't know. Okay, so you either need to know splicing very well or need to know others, because if you know splicing very well and if you know there is no way splicing adds the U, then you can easily eliminate A, B and C. Only thing left up with D. So even a person without knowing anything can answer this kind of question. So that's why I told you, it's not necessary always to know 100% of a question. Even sometimes you don't know about the question, but still can utilize your presence of mind to get an answer of a question like this one. Okay? Yes, the answer is yes. If you ask yourself this question, that can you qualify CSIR NET Life Science exam within 6 month preparation? Yes. And if you don't need to believe me, Just see the results that our students are getting who joined Shomu's biology coaching for 6-month period of time.
So I'm really thankful for Shomu's biology, and I would suggest everyone to go for it.
A lot of help and support by Suman sir always remembered in my mind.
The study materials I got helped me to understanding the topic very well, and the test series helped me to covering up the syllabus in a perfect time.
This is
And
all
also-
because of the online test series.
In-depth knowledge what Shomu's biology gave me. I want to thank Shomu biology. I want to thank each and every member of Shomu's biology for their effort, whatever they are putting on us.
And I am very much happy to choose Shomu sir for my CSIR NET GRF journey.
Really leveled up my preparation. I was confident on the day of exam.
The classes helped me a lot to understand my subject from very basics to advanced level.
So I really thanks Shomu sir and also ma'am for always guiding me.
With proper guidance, roadmap, and the help and training with perfect amount of study material and how to utilize them, the resources, everything together can help you qualify that. I am the person who will help you reach to the pinnacle of your success and your journey for the CSIR NET Life Science examination. If you trust me, if you trust my lectures, then join Shomu's Biology online coaching. Registration is going on. The details are there in the description. Fill the form to reach out to us or simply contact the number given in this video.